Hello viewers, welcome to my YouTube channel. Today I am going to explain you the digestion and absorption of carbohydrates. Generally we taking different types of nutrients in our diet like carbohydrate, proteins, fats, vitamin, minerals. These are all nutrients. But today I am only going to focus on the carbohydrates. So let's start. Before getting into the digestion part, I want to figure out about our alimentary canal, because alimentary canal is very important part of our digestive system. That part, our alimentary canal, helps us to transform the complex food material into simpler form. So digestive system is actually a system that helps the complex food material to transfer into simpler form, into simple food material that can our body absorb. Okay, so here is our alimentary canal. This is our mouth and that is the mouth cavity, and after mouth cavity there is a food pipe. That food pipe is called the esophagus. Then after esophagus there is a stomach, a bag shaped structure, a bag like structure, J shaped structure, bag like. So this is the stomach. I separate all the parts by different colors, that can you understand better. Next, after the stomach there is a orange portion, that is small intestine. Then after small intestine there is a large intestine. Large intestine is also called colon. Okay, ascending, transverse and descending colon. There are three parts of colon. Total is a large intestine. Then there is a rectum and anus. So this is our alimentary canal. Now start the digestion. Generally we take carbohydrate in the form of starch, but there are different types of carbohydrates. Overall we divide carbohydrates into three parts: monosaccharides, disaccharides and polysaccharides. Monosaccharides are the carbohydrates, that is the unit. Okay, we cannot divide these monosaccharides into simpler form. So these are the unit of the carbohydrate, monosaccharides. Disaccharides, here two monosaccharides combine to form disaccharides, and polysaccharides, here more than two monosaccharides linked by different bonds, linked by bonds. Especially here glycosidic bond is responsible for formation of the carbohydrate molecule. Here we know that glycosidic bond is help to join two monosaccharides, and peptide bond helps to join two amino acids in case figure out in the formation, in the structure of the protein. So now here today our discussion topic is, as it is carbohydrate, so we are going to focus on the carbohydrate, on the glycosidic bond. So here look, glucose and fructose, these two are the monosaccharides. They linked by glycosidic bond to form the disaccharides. So glucose and fructose, whenever glucose and fructose joined with each other, they form sucrose. Glucose and glucose form maltose, and glucose and galactose form lactose. And a long chain of glucose is a polysaccharide. So these are different types of carbohydrates, and our digestion, after digestion our body can absorb the simple form of carbohydrate, that is glucose. Okay, so let's come in detail. So as I already mentioned that the food we generally take is in the form of starch. Just for example, bread, the rice, these are all the source of starch. So starch contains, it's actually made up of two forms of glucose polymer. Okay, one is amylopectin, another one amylose. These are two form that starch contains. Look at the picture carefully. Amylopectin is a structure where we can find branch structure. Okay, but here in case of amylose form has a linear structure. A linear structure. The glucose molecules are joined by each other. They form a linear structure. Okay, and these bonds are alpha 1 to 4 glycosidic bond. This bond is responsible for formation of the amylose. This bond is responsible for the formation of glucose chain. Very clear, we say a glucose chain. These two glucose molecules attached by the alpha 1,4 glycosidic bond. But here in case of amylopectin, along with the linear chain there are branch structure, and for the branch structure another more bond is responsible, that is alpha 1,6 glycosidic bond. So for the branch structure, which bond is responsible? Alpha 1,6 glycosidic bond is responsible for the formation of the branch structure along with the linear structure. In linear structure, alpha 1,4 glycosidic bond is responsible. Okay, so two types of bond we can find here for the formation of the two forms of starch, that is amylopectin and amylose. So when we take in food, here is a bread. When we take in food, that food is breaks physically and chemically. Physically by the teeth, tongue. Okay, physically it is divided into small pieces, and then there are two salivary glands are there. Look at the picture. Three salivary glands are there, but here I am showing you two glands, and another one is at the submaxillary, submandibular and parotid. Three types of salivary glands are there. But the salivary glands actually support their secretion within the mouth cavity. Right. Within the mouth cavity, that salivary amylase that secretes the alpha amylase. Salivary, that is also called the ptyalin, that enzyme contains alpha amylase that comes in the within the mouth cavity. So this enzyme breaks the carbohydrate chemically, and our teeth, our tongue, that breaks physically. The food material is broken by by our teeth, tongue, and transform into simpler form, into small pieces. Okay. Then after mastication, when we take in food, then we masticate that food, and after mastication what happen? They form a bolus. Okay. So that breaks the carbohydrate into smaller pieces and they form the bolus. That bolus comes through the esophagus. And comes to the stomach. So within our mouth cavity, what happens? The salivary amylase breaks, that means hydrolyzes the alpha 1,4 glycosidic bonds. That bonds is hydrolyzed by the salivary amylase. Then after the esophagus, the carbohydrate with the enzyme comes into the stomach. But here, whenever they come into the stomach, they actually transform into oligosaccharides. That is the shorter polysaccharide. Shorter chain of polysaccharides. Oligosaccharides are generally which contains 2 to 7 monosaccharides. So they are actually shorter polysaccharide chain. So they are here within the stomach. We know that no carbohydrate digestion occur due to their acidic environment. That acidic environment is due to the secretion of HCl. So because of this acidic environment, the salivary amylase get inactivated. Okay. So this inactive salivary amylase couldn't able to digest the carbohydrate in further step. So here the partially digested starch present here, and the stomach pass the carbohydrate to the small intestine. Okay. So look at this picture. This is the small intestine, the lumen of the small intestine. Okay, after stomach there is a small intestine, and here is the tubular part, that is the small intestine, and here is the lumen. I want to notice one thing here. This is our pancreas. Okay, this is our pancreas. Pancreas. From the pancreas, pancreatic amylase that secretes into the small intestine. And that pancreatic amylase again hydrolyzes the rest of the alpha 1,4 bond. Here it is. Okay. Now I am going to show you one structure. At the lining of our small intestine, as we know that our small intestine have finger-like projections, that for the projections are called villi. Also we can call them as a brush border. Okay, so this is the brush borders or the villi, and these cells is called the enterocyte. These cells are called enterocyte. And with the enterocyte, they are lined by blood vessels. Blood vessels are after that enterocytes. Blood vessels are present after the enterocytes. Actually, they surrounds the small intestine. That can the food absorbed and come into the blood. Okay, so first look at the picture here. There are different enzymes that called the brush border enzymes. Okay, there are some enzymes called brush border enzyme. That enzymes are maltose and sucrase. Maltase and sucrase. So here it is the maltase, sucrase, and this is sucrase, and also we can say isomaltase. So when this part of carbohydrate, that is the shorter chain of the oligosaccharides come into the small intestine. So after coming into the small intestine, there are other enzymes that breaks that oligosaccharides into simpler form. What happened? The maltase hydrolyzes the maltase, and sucrase hydrolyzes the alpha 1,4 and also alpha 1,6 glycosidic bond. Okay, sucrase and isomaltose. These are the two enzymes. Maltose hydrolyzes the maltose, and sucrase or isomaltase hydrolyzes not only the alpha 1,4 bond, also alpha 1,6 glycosidic bond. As they act on the alpha 1,6 glycosidic bond, that means the breaks into linear chain. This branch breaks into linear chain of glucose. Okay. Now what happened? These enterocytes enzymes, these blue portions are the maltase, these are sucrase. They breaks the carbohydrate into simpler form, that means into glucose. Now I am showing you on my next slide. Look at this picture very carefully. These are the monomers. After breaking down by the two enzymes, maltase and sucrase, the oligosaccharide chain transformed into simpler glucose molecule. Okay, then in the lumen of the small intestine, their glucose molecules, the number of glucose molecule increased. Now it needs to be absorbed. So how they absorb? They absorb like that. One more thing is present here, that is sodium ions. Sodium ions are also in the lumen of the small intestine. Only glucose cannot be passed into the enterocyte. They need 2 sodium with them. So by the channel, the name of this channel is blue portion, the blue channel, that is SGLT. SGLT, the full form of SGLT is sodium glucose linked transporter. Okay, that helps one glucose molecule and 2 sodium molecules to come in the enterocyte. Okay, this chain helps the two sodium ions and one glucose molecule to enter within the enterocyte. Now, another type of carbohydrate, that is the fructose. Fructose can come through this channel. The name of this channel is GLUT5. GLUT5. That channel helps the sucrose to come in the enterocyte. And galactose can come through this channel, SGLT. So SGLT is a channel that transport the glucose as well as galactose. And GLUT5. Okay, this channel helps the fructose to come in the enterocyte. Now, in these cells, in the enterocyte, their glucose comes. Okay, sodium may transported at the back opposite direction by this channel, but glucose ions get the number of glucose ion getting increased within the enterocyte. Now what happened? There are another channels are there. These green portion, I mark them by green color. So these channels called GLUT2. Okay, GLUT2, that help the glucose molecules to come within the blood cells. That means the glucose get absorbed within the blood. Okay, then this is the lumen of the small intestine. Lumen of small intestine. This is the lumen of small intestine. So from the lumen of the small intestine, the glucose molecule get entered within the blood vessels. First by the SGLT, then from the enterocyte by the GLUT, ultimately comes to the blood vessels. Now the blood is full of glucose molecules. Now what happened? When the glucose molecules come within the blood, they may store as a liver glycogen. They may store as a liver glycogen. They may become a part of our blood glucose. May store in our muscle as a glycogen, or may converted or stored as a fat. So these are different activities may happen whenever glucose come in our blood. And another two carbohydrates, that is fructose and galactose, these are also absorbed within our blood cells, within our blood capillaries, within our blood. So that also may convert it into glucose. So glucose is actually the simpler form of a carbohydrate that our body can absorb. Apart from the glucose, there are some other form of carbohydrates that we call some chains. Chains of carbohydrate that are not absorbed by the small intestine. They remain undigested. That undigested chain of carbohydrate, that is called the resistant starch. They are actually not able to absorb by the small intestine. Rather, we can say they are not able to digest by our enzymes. So that form of carbohydrate, that form of carbohydrate is called resistant starch. That starch come to the small intestine. From the small intestine, they come to the large intestine. So now what happened with the resistant starch within the large intestine? Let's see. Here is the lumen of our large intestine, that is also called colon. What happened? This is resistant starch. Here some bacteria are present. We call them as a gut bacteria or a gut bacteriophyta. So that bacteriota or biota, microbiota often called. So this bacteria, by the fermentation, they transform the resistant starch into short chain of fatty acids. That fatty acids may get absorbed by the cells of the large intestine. Here some mucus are also there, they secrete mucus, so they get absorbed by that part. Okay, so this is the fate of the resistant starch. I have done a mistake here, it would be starch, C-H, starch. So, whenever these resistant starch get utilized by our body, and there are some other parts that are not take part in digestion, that is actually excreted from the human body, that actually come to the rectum, and at the time of excretion, at the time of defecation, that fecal matter, they form a fecal matter here, and then in certain time they get out of the body, that we call the defecation. The process of the discharge of fecal matter we call as a defecation. So we look that how our digestion, carbohydrate digestion, starts from the mouth and it ends up to the rectum or anus. So this is all about the digestion and absorption of carbohydrate. I think I cleared you well. If you have any doubt, any questions regarding this topic, then definitely ask me in my comment section. And if this video help you, then please give a like and definitely share this video with your friends. And if you are new in my channel, then don't forget to subscribe. I will come very soon with my new video, new topic. Till then, take care. Thank you for watching.